So in this video, I explain about the types of structural types of immunoglobulins. The immunoglobulins are the glycoprotein. Immunoglobulins are hormones and antibodies are most of them are protein in nature. Immunoglobulins are a glycoprotein. Globulin is a type of protein. Globulin, albumin is a type of protein. So this immunoglobulin, why it is called as immunoglobulin means because it is involved in the immune response. So it is called immunoglobulin. All immunoglobulins are not antibodies, but antibodies are immunoglobulins. All immunoglobulins are not antibodies, but antibodies are immunoglobulins. These immunoglobulins have 5 different types of heavy chain. It has heavy chain and light chain. So in heavy chain there are 5 different types. Based on this type, it is classified into 5 different types: IgA, IgM, IgG, IgD, and IgE. So in the structure of antibody, there is a heavy chain and a light chain, 2 identical heavy chain and 2 identical light chain. So in heavy chain, there are 3 different region. One is constant, 3 constant region and 1 variable region. In light chain, there is 2 region, one is constant region and one is variable region. Constant region is remains same, it is constant for each and every antibodies, but variable region, it will be varied based on the amino acid sequence. Each and every antibody has different amino acid sequence in their variable region. So it is called as variable region. So these regions are joined together by a disulfide linkage. These regions are joined together by a disulfide linkage. Among each heavy chain and each light chains are joined together by a disulfide linkage. So it's like, it looks Y-shaped in structure. So in this region, the upper portion is called as Fab region. The upper portion is called as Fab region. What is the expansion of Fab? Fragment of antigen binding. So it is also called as paratope. It is also called as paratope. This paratope region is binds with the epitope region of antigens. So this region is called as paratope where the antigen binds. So each and every immunoglobulin has 2 different antigen binding site, Fab region, 2 different Fab region. The lower portion is called as fragment of crystallization or FC region. So it is the effector region which crystallize the antibodies. So the upper portion is called Fab region where it contains 1 constant heavy chain and 1 variable heavy chain, 1 constant light chain, 1 variable light chain where the amino acids, where the antigens are binding. So in Fab region, it is also called as paratope. In the FC region, it is fragment of crystallization. It is called as effector region. So in immunoglobulins, the end, one end has amino region, another end has a carboxyl chain. Because it is a protein, obviously it contains amino acid. So the one end will be amino terminal end, and the other end will be the carboxyl terminal end. So the structure of immunoglobulins. The immunoglobulins are in case of heavy chain, it is it has 5 different types. That is alpha, gamma, lambda, mu, epsilon. In case of light chain, there is 2 types of light chain, kappa and lambda. So in each and every immunoglobulins, there is an identical heavy chain or identical light chain. That is, in case mu chain means it is mu chain. 2 chains are mu chains. It is epsilon chain means it is 2 chains are epsilon chain. And at the same time, the light chains are also same chain. It is 2 chains are kappa means there are 2 chains are kappa. Does not has 1 kappa chain and 1 lambda chain. It has 2 kappa chain or 2 lambda chain. In case of heavy chain, it has 2 mu chain, 2 epsilon chain. So based on the heavy chain, it is classified into 5 different types. The fragment Fab region, it has 2 Fab region. In case of different types of immunoglobulins, for example, IgM is a pentamer. So 5 immunoglobulins are joined together by a chain. That has 10 different Fab region. So it is differ based on the antibody, but a common structure, it has 2 different Fab region where the antigen recognize and binds. In case of FC region, it is called as effector region where the antigens will be processed, where the processing of antigen takes place and it binds with the another cells. That is called phagocytic cell, opsonization process, cell receptors. When where the complete the complete eradication of antigens will be takes place, but the process of antigens should be takes place only in the FC region. So there are 5 different types of immunoglobulins: IgG, IgA, IgM, IgD, and IgG, IgA, IgM, IgD, and IgE. So IgG immunoglobulin is the most abundant immunoglobulins in our body. So it is the only one antibody which is transferred through the which is transferred to the mother from baby while the baby is inside our womb. Even the baby is inside the womb, the immunoglobulin will be transferred from mother to baby. So baby's blood count as baby's blood has more abundant amount of IgG. So this is the structure of IgG. Then IgA. So this is the second abundant immunoglobulin. So this IgA is the dimer. So there are two different immunoglobulins. There are two immunoglobulins are joined together by a chain. So it is a dimer. The half-life period will be 6 to 8 days. So each and every immunoglobulin have different half-life period. So this is the structure of IgA. There are two immunoglobulin structure. The two immunoglobulins are joined together by a J chain, joining chain. Then IgM. So this immunoglobulin IgM is a pentamer. There are 5 different types of immunoglobulins. Each immunoglobulins have mu type of heavy chain, and it may be the light chain may be either kappa or lambda. So these all 5 chains are joined together by J chain. So there are 10 different Fab region here. Then 10 different Fab region and 5 different FC region. Then IgE, so it is an antibody which is responsible for allergic reaction. Then IgD, the function of IgD is not known. The concentration will be given in the slide. So it protects the each and every immunoglobulins. What is the role of the immunoglobulins will be displayed here. The function of IgD is not known. IgE unit is responsible for hypersensitivity reaction. Ig blood health, plasma health, then body fluids, it protects the body fluids also. So this is the overall view of the functions of 5 different immunoglobulins. Thank you.